Okay, if you watch the recording of this, I didn't pause the recording for about 5 minutes after I logged on. So there should be a few minutes of absolutely dead air before anything happens. And so I'm going to start chapter two. And chapter two is a very long chapter, and it should be a review. And it's such a review, I'm actually going to skip a lot of chapter two. I'm going to skip the mitosis part. I'm going to assume that you remember that from Biology 1033. If I need to go back and cover it, I can. I have also, let's see, not what I want to share. It's not what I want to share. That is what I want to share. I have also posted homework. And so if you go to the homepage, and I will switch over to the student view. And so to get the homework, you go to assignments. And I have posted this cell cycle worksheet that you click on, and you'll have to download it. And you'll save it somewhere on your computer. And if any of you took me for Biology 1, you've already done this. And if you took somebody else for Biology 1, you've probably already done this. And so, there's some reading, and there's a bunch of questions, 30 odd of them. And you don't have to worry anything about this part. And after you've answered the questions, you'll have to save the document with your answers on it. And you can go back to that assignment. And you'll have to upload it. And you say, here, choose a file, go to where you saved it. And submit. And, the confetti and whatnot. And then if I leave the student view, I can go to my grade book. And I, by the way, am the test student. And so you get a little icon in the cell cycle, and it should be by your name. And you have a week to do it. And so today's the 22nd. And you have until next Thursday to do it. It shouldn't take more than a couple of minutes to actually do. And I went ahead and just made it worth 10 points. I just had to pick a number, so I picked a number. And that is where I want to start. That's the file I want to share anyway. And so this is chromosome transmission and reproduction. It's the cell cycle. That's stuff you've already done. And so there's some, basic definitions. Reproduction is just the way in which new cells or organisms are made. It does require the transmission of chromosomes from parent offspring. And especially for eukaryotic cells, the chromosome must be sorted because most eukaryotes have multiple chromosomes. And so each cell must get the appropriate number in combination of chromosomes. And it goes on and it defines a chromosome. And this is all, review. Chromosomes are just the structure that contain the genetic material. It should be reviewed. They're composed of, especially true in a eukaryote, they're composed of DNA and proteins. And the proteins are very important in a eukaryotic cell because it allows the DNA to be compacted enough to fit inside the nucleus of that cell. And that combination molecule is called chromatin. And okay, two types of cells, prokaryotes and eukaryotes. We're not going to worry too much about prokaryotes. If I find anything about prokaryotes I care to mention, I'll stop and I'll mention it. I'm not going to have any pictures on the exam to identify parts of the cell. And so here's a eukaryotic cell. It's got a nucleus, got a bunch of chromatin inside it, and a variety of other organelles in the cytoplasm. And again, everything here should be a review. During some phases of the cell cycle, the chromosomes can actually be distinguished with a light microscope. Every chromosome has a certain size, it has a certain staining pattern, and it has a certain shape. And the karyotype is simply the image of all the chromosomes found in that organism's body. And if you want to know how to make a karyotype, you can. It's really, really kind of boring. Now, most eukaryotic species have two copies of every chromosome. And the number of chromosomes is going to vary from species to species. And it's not really indicative of anything other than the size of the chromosomes. And so dogs, for example, 78 chromosomes. The chromosomes tend to be small. Fruit flies have 8 total chromosomes. They tend to be relatively large. And I'm not going to go through binary fission. And I'm not really going to go through too much of the eukaryotic cell cycle. You should remember that you can divide the eukaryotic cell cycle into interphase and M phase. And interphase is collectively G1S and G2. And then M is mitosis, which is division of the nucleus. And, there it is. And it's also on that worksheet. I'm not going to go through restriction points, and I'm not going to worry too much about any kind of thing going on in S, other than I'm going to point out that these terms in bold are new to genetics. In the case of phase S, the chromosomes are replicated, and the two chromatids are joined at the centromere, and collectively you refer to that as the dyad. And there is a special place in the centromere called kinetochore. And the purpose of the kinetochore is to help sort the chromosomes during division. And basically, what your cells can do is they can count the number of kinetochores they have. And when they count the number of kinetochores, they count the number of chromosomes. And now I'm not going to through what happens in G2 phase, and I'm not going to go through what happens like in G0 phase. Cells that never, ever, ever prepare to divide again go into G0, and they can stay there for a lifetime if it's necessary. And then I'm going to say, well, there is a mitotic spindle. In animals, you have a pair of centrioles, and they make up the centrosome. Plants and fungi and other eukaryotic organisms have to do without these centrosomes, but they do have these things called microtubule organizing centers. And I'm not going to go basically through all of the microtubules you have, but there are 3 different types. Some attach to the kinetochore, some grow backwards and make the cell elongate. And some are what they call polar microtubules. They simply guide the development of the other microtubules. But I do want to get a little bit with meiosis because it goes into a little bit more detail. If the typical eukaryotic cell is diploid, the results of meiosis is it makes a haploid cell. It makes a gamete. It makes a sperm cell or an egg cell. Just like in mitosis, meiosis begins after the cell has gone through G1, S, and G2. And you have meiosis 1 and meiosis 2. And we can not deal with prometaphase. So the events of prometaphase, we're going to say they occur in prophase. And so there's prophase 1, metaphase 1, anaphase 1, and then prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, in prophase 1, and this is where it gets a little bit more detailed than in general biology. Prophase 1 can be further divided, and you can divide prophase 1 into something called leptotene, zygotene, pachytene, diplotene, and then there's diakinesis. And there is pictures what happens in leptotene, the first part of prophase 1. In zygotene, you begin to have the homologous pairs line up. By pachytene, this is completed. In diplotene, you see the formation of what they call the chiasma, the crossing over event. In diakinesis, the nuclear envelope is completely broken apart, and that exchange of genetic material occurs. And so the first few stages in leptotene, zygotene, and pachytene. In leptotene, chromosomes begin to condense. They begin to become visible. In zygotene, the homologous chromosomes align and they form that synaptonemal complex, basically the crossing over. In pachytene, the alignment is complete. And in pachytene, what you have is you have pairs of sister chromatids. So 4 chromatids in total, making up something you can call a bivalent, because each one is 2 chromosomes, or a tetrad, because each of these things is 4 chromatids. And at that stage, the crossing over occurs. The physical flopping over of pieces of the chromosome occurs. And then you have the crossover, and crossover involves a physical exchange of genetic material that results in you basically shuffling the combination of genes on each chromosome. The crossing over event might happen more than once per chromosome, and it can happen once or twice or several times. And the size of the chromosomes determines how often, how many crossover events happen. Um, the species helps determine how many crossover events happen. And for example, during human sperm formation, this crossover occurs about twice on average. And during this crossing over, you form a chiasma, which just means it's a cross. And then in diplotene, the complex, the synaptomere complex, has mostly vanished. It's held together by a bunch of proteins, and these proteins that form this complex get enzymatically broken down. The chromatids, since they're not held tightly together, move a little bit away from each other, and at the end, that synaptomere complex completely vanishes. And I'm just going to say this is the rest of prophase. The chromosomal pairing and crossing over are done by the end of prophase. Technically, in metaphase 1, you see the formation of those spindle fibers. And the chromatids are completely attached to those spindles via the kinetic or microtubules. And so here is, and then they go early prophase, late prophase, and then, okay, metaphase. And then the rest of the phases of meiosis 1. And so here it is, metaphase 1, chromosomes align at the equator of the cell. Anaphase 1, the paired bivalents pull away from each other. And then there is a round of cell division. So in metaphase 1, the bivalents are organized along the equator of the cell, the metaphase plate. It just means the middle of the cell. If you looked at a picture of a cell in metaphase 1, the chromosomes should be in a double file line. Now, other than the fact that the homologous chromosomes must be paired, the arrangement, the pattern in which they line up at the middle of the cell is completely random. And so the more chromosomes you have, the more chromosomes a species has, the more possible arrangements can be made in metaphase 1. And so the mathematical, and that N should be a superscript. In a diploid organism, there are 2 to the N possible combinations of alignment in metaphase 1. Humans have 23 pair of chromosomes. And so therefore, in a human, there are 2 to the 23rd possible combinations of alignment of the chromosomes. And if you do the math, I think 2 to the 23 is 8.4 something million. And so there are 8.5 million possible ways chromosomes can line up in meiosis 1. Another big difference from mitosis, one pair of the sister chromatids is linked to one pole. The other pair is linked to the opposite pole. Compare that to mitosis. One pair of sister chromosomes is linked to both poles because you're about to go into metaphase, and that pulls the replicated chromosomes away from each other. And so in metaphase 1, you have one pair of that bivalent linked to one pole, one pair linked to the other. And, that color combination shows that crossing over has occurred, and the genetic recombination of the genes on these chromosomes has also occurred. In anaphase 1, the 2 pair of sister chromatids separate from each other, but in anaphase 1, the sister chromatids are still joined at the centromere. And so basically in anaphase 1, what you have done is you have split the dyads apart. You've cut the tetrad into a pair of sister chromatids. In telophase 1, those dyads have completely separated. They've moved to opposite poles. At this point, The chromosomes may decondense. The nuclear membrane might reform. Not necessarily. You could segue directly into meiosis two, and there's no pause for this to happen. Again, it depends upon the species. It depends upon what kind of gamete is being made. At the end of meiosis one, you should have two cells. Each is going to be haploid, but the amount of DNA present in those two cells that is made is still twice as much as there needs to be in the end product of the gamete. And you can refer to meiosis one as reduction division because you've reduced the chromosome number from a diploid number to a haploid number. And then you go into meiosis two. And meiosis two is pretty much exactly the same as mitosis. Only major difference is that when you start meiosis two, the cells are already haploid. And for example, do the numbers. Assume that an organism has 6 chromosomes. Mitosis is going to begin with 12 chromatids joined together as 6 pair of chromatids. Compare that to meiosis 2, you have 6 chromatids as 3 pair of sisters. And then meiosis 2 is going to go through prophase and I guess prometaphase. And then metaphase, anaphase, and telophase. Except now you have to put the Roman numeral 2 after each phase of meiosis 2. And so the big difference is that you look and in metaphase 1, you have a double file line. And in metaphase 2, you're back down to a single file line. And then you proceed through anaphase and telophase. Now, and again, assuming that the eukaryotic organism you're referring to is a diploid, mitosis should make 2 diploid daughter cells that are genetically identical to each other, and they are genetically identical to the mother cell. And we tell a little lie at this point. And we say that meiosis, I got to stop the sharing it. There. I don't know why I want to do that, but there. That makes me feel a little bit better. Okay. Say the lie and say meiosis makes 4 haploid daughter cells. And the events of meiosis, that random alignment of chromosomes in metaphase 1 and the crossing over that occurs in prophase 1, ensures that all of those cells that are produced are not identical to each other. And they are not going to be identical to the cell that started off the division. And the daughter cells are only going to have 1 homologous chromosome from every pair. That's one big difference between the mother and the daughter. And the daughter cells can contain all kinds of different combinations of chromosomes. And moreover, they can contain all kinds of different combinations of genes that are found on every chromosome. And so basically, and Robert Brooker loves tables. Here's the event. Does it occur in mitosis, meiosis 1, or meiosis 2? And so you have synapses, and followed by crossing over. And then Does this synapses happen in mitosis? No. Does crossing over happen in mitosis? It can. Does crossing over happen in meiosis one? It says commonly, as far as I know, it always does. Does synapses happen in meiosis one? It has to. In the prometaphase step, the sister chromatids in mitosis better be attached to both poles. And in meiosis one, they're attached to one or the other. In metaphase, the sister chromatid line up, single file line along the equator. In meiosis one, they line up in bivalent. Anaphase, in mitosis, the sister chromatids separate. In meiosis one, it's the bivalents that separate. And when you compare mitosis to meiosis two, basically it's the same thing, except at the end of meiosis two, you've made haploid cells, and you've made at least 4 of them. Now, there are a couple of types of reproduction: sexual reproduction and asexual reproduction. And then we go right into the animal kingdom, and we go into the plant kingdom for a little bit, and we don't discuss the fungal kingdom or the protist kingdom at all. And so sexual reproduction is actually common in the eukaryotic domain. And what happens in sexual reproduction is the parent organisms have to make gametes. These gametes better have half the amount of genetic material found in the somatic cells of the parent organism. There has to be a fusion, a cellular fusion between these gametes of opposite types. And then there's going to be a nuclear fusion to go back up to the diploid number of chromosomes. And the process of making gametes is simply gametogenesis. And so gametes have, typically, we say they're haploid, a single set of chromosomes. And the abbreviation for that, haploid is abbreviated 1N, diploid is abbreviated 2N, whereas N is the number of chromosomes. And so in humans, diploid cell has 46 chromosomes, or it should. And a human gamete should only have 23 chromosomes. You know, if everything goes right, it has 23. And so anything capable of sexual reproduction has cells in its body somewhere that are capable of dividing via meiosis. And you make haploid cells as a result. And the cells have to be careful to sort the chromosomes out correctly. And it turns out that all kinds of errors can occur. This sorting can be done incorrectly. And the thing is, in mitosis, which produces typically somatic cells, all of the genes in those cells are expressed. And if chromosome number is abnormal, it causes problems, usually causing the death of those cells. Gametes don't express the genes. And so if they have an abnormal number of chromosomes, they can survive. And they can contribute an abnormal chromosome number to the next generation. And that can cause some problems. Now, if you really want to know, some eukaryotic species are what they call isogamous. That is to say, the gametes that are produced are about the same size, and they look a lot alike. And they typically are more simple eukaryotes, representatives of the fungal kingdom and certain protist organisms, certain algae. Most species are heterogamous, and the cells that are produced are strikingly different. And typically, we just say the sperm cells are the male gamete, and the sperm cell is very small, and it has a flagellum. It can move under its own power, and the egg cell is not mobile and is usually much, much, much larger than the sperm cell. You have inherited all of your organelles from your mother. Your father has only contributed half of a genome to you. So spermatogenesis is the production of sperm. And male animals, it occurs in the testes. And you have certain cell types. And you have a spermatogonial cell that divides via mitosis. One of those cells that is made is going to remain a spermatogonial cell. The other one will develop into a spermatocyte. That spermatocyte goes through meiosis 1 and meiosis 2. And so meiosis 1 yields secondary spermatocytes. They're haploid. Meiosis 2 yields spermatids, which is basically an immature sperm. And each spermatid has to then mature into the gamete. Basically, it develops a flagellum and it packs a whole bunch of mitochondria into what they call a little mid piece here to enable it to travel. Now, the structure of a sperm is pretty simple. It's got a long flagellum and it's got a head. The head contains the nucleus. There is a little cap on the head called an acrosome. And the acrosome contains enzymes. And when the sperm fuses with an egg, it's those enzymes that digest their way through the egg cell membrane, and the nucleus of that sperm then enters the egg cell. And in the case of animals, the fusion of the two nuclei is basically done immediately after the two different cell types meet. In human males, at least, spermatogenesis basically, and I say this to my biology 1 students all the time, basically, spermatogenesis begins at puberty and it continues until death. And so the typical male might produce several 100 million sperm a day. Oogenesis is production of egg cells. And animals, it occurs in the ovaries. Early in development, the oogonia are going to divide and make oocytes. And the number of oocytes differ from species to species. But in humans, you produce about 1 million of them. Those oocytes start meiosis 1, and then they stop. They stop in prophase 1. And then at sexual maturity, some of them will go continue through meiosis. And the number of oocytes that continue depends on the species. And so at maturity, every now and then, those primary oocytes are activated. They finish meiosis 1. In cytokinesis, at the end of meiosis 1, the cell division is not equal. It makes two cells one of which is basically a nucleus. It's a polar body. It dies, and it makes what they call the secondary oocyte. If that secondary oocyte meets the sperm cell, it very rapidly continues through meiosis two. And again, it makes, well, it gets fertilized and makes a zygote, and it makes another polar body. And so in egg production, we say that, well, in egg production, only one of the four cells that can potentially be made through meiosis becomes an egg. Now, secondary oocyte starts off meiosis two, but stops. And in the typical mammal pattern, at least, that oocyte is released into the oviduct. That is ovulation. If that secondary oocyte meets the sperm, it is fertilized. It rapidly goes through meiosis two, and you get an egg produced and another polar body produced. And then in animals, the nuclei of the egg cell and sperm cell fuse almost immediately after the cells fuse, and you get a diploid zygote, which develops and becomes, you know, a new animal. And so here's the production of polar bodies, illustrated by this. Basically, it's a nucleus. Now, plants. To make life for biology 2 students difficult, plants have what they call an alternation of generations. And they can alternate between haploid and diploid. And the haploid generation is called the gametophyte. It makes gametes. And the diploid generation is called the sporophyte. It makes spores, which germinate to become the gamete, the gametophyte, which makes gametes. And so plants do this in a way to irritate biology 2 students. And so in plants, meiosis makes these things called spores. Spores are going to divide mitotically, and they will develop the gametophyte generation. The general trend of plant evolution. Very simple plants, like the moss. We refer to mosses and related plants as gametophyte dominant. And so if you see a moss, the free living plant that you observe is the gametophyte. And they have relatively high cell counts. As you go further through plant division, you go in from mosses, you go into the ferns, and into the gymnosperms, and finally into the flowering plants. What happens as you go through plant evolution, the gametophytes get greatly reduced. And so by the time you get to the flowering plant, the gametophyte is almost, if not microscopic. And it is completely dependent upon the sporophyte that produced it. And so in flowering plants and in gymnosperms, in pine trees, for example, and in any flowering plant, when you see the plant as an organism, that is the sporophyte. And then we say that those plants are sporophyte dominant. Now, in flowering plants and non-flowering plants, this is a little bit different. But in a typical flowering plant, and this is the typical flowering plant, you have several organs in a flower. And you have these things called sepals. They're very leaf-like. They are the outermost part of the flower. Until the flower opens, they encase the flower. And then when the flower opens, you see the petals. And the petals are often the colored part. They make any odor the plant might have. And then you have the male gametophyte, which are the anthers. And they end up making the pollen grain. And then you have the ovary, which is going to make the female gametophyte, which is called the embryo sac. And in the case of a flowering plant, this embryo sac is going to be encased by the ovule, which will eventually mature into the fruit of the plant. Now, plants versus animals. Animals make gametes by meiosis. Plants make spores by meiosis. And these spores develop into a gametophyte. And these gametophytes develop gametes through mitosis. And in that gametophyte, there are certain cells that are going to mature into the plant gamete. And I'm hoping that I skipped mitosis. I'm hoping it would be a review. If I need to go back through and cover the steps of mitosis, let me know, and I will sit down and do a Zoom session and record it and post my mitosis work. Now, I have another assignment that's looking at pictures of cells or reading a description of what's going on in a cell and saying, is that picture showing a cell that's in metaphase 1? Is that picture showing a cell that's in anaphase 2? Or when I read this description of where the chromosomes are, is that indicative of something in prophase 1 or whatever? I have not posted that. I can if the need arises. But for either one, if you feel the need to look at that homework, or if you feel the need for me to go over mitosis, let me know, and I'll post everything on Canvas. I don't have anything posted on Connect yet, and I don't know if I will on the early chapters, because I really don't like the way the homework is structured for this textbook on Connect. It's just, since this is an online class, it's easy to post homework virtually and have it graded automatically. And so I'm going to stop this. I'll get the recording in a half hour or so. I'll post it. I'll email it to everybody. And remember, at the beginning of this recording, I have about 5, 6, 7 minutes of nothingness and are doing some construction, building a new building on campus right outside my office window. And I think the construction noises might get recorded as well. And so if you hear random things, that's someone building a new building outside. Well, it's been 45 minutes, long enough. At the end of chapter 2, and I will do chapter 3 and 4 next week. And I looked at the schedule earlier. Exam 1 is scheduled to be on or around February 6th, which is the end of the first week of February. and I should finish easily by then. By the way, I don't know if this has any effect on you or not. School might be closed Monday morning. It depends upon how late that rain comes in on Sunday. But they're forecasting a little bit of ice buildup over the weekend.